introduce our speaker for today.
Good morning, everyone. I am Mrs. Amrita Bhatia. Take this golden opportunity to introduce the guest speaker for technical session 1, Dr. Jyotika Rajpuri, Scientist Head, Nanobioscience Group, ARI, Pune. Ma'am is a distinguished scientist specializing in microbiology, biotechnology, and nanoscience applications in various fields. She completed PhD in microbiology from Pune University. Ma'am is recognized as a guide for MSc and PhD students in microbiology as well as biotechnology and Pune University. She has an extensive research experience, having worked as a research associate with CSIR and Department of Biotechnology at Pune University. Ma'am has also worked as project scientist and research scientist at ARI. She has been a part of Nanobioscience Group since 2007. And in addition to her research, Ma'am is actively involved in several professional societies, including BRSI, ISCB, and AMI. She also contributes to academic governance, serving on various advisory and research boards, including Symbiosis University, Pune University, and North Maharashtra University. Ma'am is also a member of selection committee for ESC's Vice Scope Fellowship Program under Vice Pillar Division. She has authored over 55 research papers, contributed to 10 book chapters, holds 10 patents, and has mentored 12 PhD awardees and 3 postdoctoral fellows. Ma'am is recognized as an expert in her field with over 35 abstracts accepted at conferences and more than 40 invited lectures and oral presentations. We are really honored to have such an eminent scientist amongst us for our today's event. I welcome Ma'am on behalf of the Olympic Committee. I now request Ma'am to take over the session and start with her talk.
Before ma'am starts her talk, I request Mrs. Amrita Bhatia to facilitate our guest for today as a token of our love and appreciation. I request ma'am to start her talk. Thank you, Amrita ma'am. Thank you, ma'am.
I think I'm audible. First, let me thank Dr. Mujumdar. She is my batchmate. We studied together at MSc, and since then, we have been in close contact with each other. Due to the Cognizant pandemic, I have not visited this campus since 2018. But I'm happy to be back here, and I have seen there is huge development in the campus also. So that itself tells that about the enthusiasm of not only the students but your professors who are pushing you to your limits to get the most out of you and to make you learned scholars. Uh, I begin my actually the theme of this particular conference is also very interesting because all of us are seeing degradation of the environment to a very large extent, and it's really our responsibility to take informed and proper decisions in order to
save something for the future generations. And with this head in mind, I have tried to focus my little research contribution, and I will present that small piece of work with you. There could be some slides where we are talking about or discussing the context of bacteria in the context of bacterial cellulose. The recent advancements in plant cellulose, it is always associated with substances like hemicellulose, but that's not the case with this particular cellulose. And certain bacteria producing really copious quantities, like gram per liter, and up to 8 to 10 gram per liter is the heat of cellulose. And in PPT, download. She has got here to take up some control to that. The girls are amazing and her work goes really. She also taken two book chapters. We have two patents to her credit. She has been invited to various conferences for talk, answer, intellectual, and the latest. She has also attended young investigators. She has worked at scientific field of games and research. She has served as a reviewer, manuscript, journals, scientific reports. She is a member of the technical committee on biomedical. She has undertaken many projects in India Council of Medical Research and India.
Thank you, ma'am. I would now request our guest, Dr. Deepa ma'am, to start her talk.
Good morning, everyone, and first of all, thank you so very much for inviting me here and for such a really kind introduction. It's a pleasure to come in. It's a beautiful hall. I just love the gallery there, the site, the selling and it's really beautiful. I like the acoustics a lot. So, without further ado, let me tell you a little about what we do today. I was told that there are bachelor's, master's, and PhD students, no faculty programs. But if at any point of time I don't make sense, please raise your hand to stop me. Okay? So, as Nan mentioned, I'm the National Center for Cell Science, which is a research institute which is inside the university campus. I think some of you have visited the institute at various times. Apparently, I've also taught people during coursework. I apologize if I don't remember your names and faces. So, when we think of the human body, we think of the body plan of the organism. What you notice right at the beginning is that there are so many different cell types, so many different structures, and each one is so distinct from one another. But then they all come together to form this amazing functional organism. This can be anything from a worm to a fly, to a human being, to a mouse, to a dog, to an elephant. All of them have this variety of tissues and organ systems which all work together to form a functional organism. So the question then starts is how does one build an organism? How do cells within the organism know what to do and where to do? And when things go wrong in processes which occur within cells, how does the cell suffer and how does the organism deal with it? So this is sort of a very large question that we try to address in the lab and that we work on. So if you think about early development, if you think of vertebrate development, life starts as an embryo, right? A fertilized embryo where the egg is fertilized by the sperm. And this single cell structure, called a zygote, then undergoes division to give rise to the 2-cell stage, the 4-cell stage, the 8-cell stage, a ball of cells called the volumina, which then further cavitates and forms the structure called the blastocyst. Judging by the rods in the audience, I think you all know this, which is excellent. So this particular set of cells which occurs towards the inside of the blastocyst is called the inner cell mass. And this bundle of cells, it's around 8 to 20 cells, this is what gives rise to every single one of us. In other words, it's a very humbling thought to think that something microscopic, just a handful of cells, less than 20 cells, have given rise to a complex organism which can walk, which can talk, which can think, which can carry out physiological functions. All of those functions come from this tiny little pool of cells which occurs here. How does one study this? How does one understand how development occurs? In order to do this, that means each time you have to take an embryo, you have to start with an embryo, which means that the embryo cannot be formed to form the organism it needs to form. If you want to study what are the developmental processes which happen, what are the genes which are functioning, what are the proteins which are functioning in that. So one way to overcome this problem is you can choose a few embryos, you can isolate these cells from the inner cell mass, and you can maintain them in culture. Once these cells are in culture, in the right culture media, they will stay forever. If you maintain them correctly, they will stay in that state forever, multiplying and producing more cells, and retaining that state of the cell. And such cells in culture, which are isolated from the inner cell mass, and which retain the properties of the cells in the inner cell mass, in culture, they are called embryonic stem cells. And this is precisely the model system that we use in the lab to try and understand early aspects of mammalian development. So how do we use stem cells and why should we use stem cells? So the beauty of embryonic stem cells particularly is that once you maintain them in culture, they respond to stimuli just as how the cells within the embryo in early development do. And these embryonic stem cells then go on to form all the cell types in the body, whether they're pancreatic cells, hematopoietic, cardiomyocytes, neurons, hepatocytes, epithelial cells, this goes on. So now here you have a system which, in a dish, you can take these cells and you can sort of tell them what to become, and now you can start looking at various things which are going on in the cells, for example, gene expression, you can look at protein expression, you can look at RNA, modifications, whatever you want to. But now you have a system where you're not continually taking an embryo and destroying it, but you've taken a set of cells which have been formed from an embryo and you're using them to address questions pertaining to the development. So, just to sort of make sure we're all on the same page, stem cells have two main properties. One, they can all self-renew, which means that one stem cell can form two identical stem cells, and so you will increase the number of stem cells without losing any of the properties of stem cells. The second thing that stem cells can do is that they can differentiate. And differentiate, as the word itself suggests, means to form something slightly different from the cell, which means a stem cell, in response to a specific signal, in response to a specific cue, can form a cell type which is very specialized, which has a defined function, and it can differentiate itself from a stem cell. It can form a cell different from a stem cell. So two problems: stemness as well as differentiation. Another word for stemness is pluripotency, which is the ability to form cells of all three germinates. Okay. So with this background information, let's move on. So here is just what I told you about self-renewal or symmetric division, that if you have a stem cell which is marked here in yellow, the stem cell can divide and form two identical cells which are stem cell-like. So now you are just increasing the population of stem cells without creating a different type of cell. Now the other thing that I told you about, differentiation, that is here. So you can have a stem cell which asymmetrically divides, that when it divides, one of the daughter cells goes on to form a stem cell, which will then maintain your pool of stem cells, and the other cell that you have, it will form a differentiated cell. And this can happen in a variety of ways. This cell can either be a progenitor, or then you go on to have a few more divisions and produce your differentiated cell, or you can take different parts. But suffice it to say that in this mode of division, you will get one stem cell and one non-stem cell. So stem cell types, just to sort of summarize, three main stem cell types are usually talked about. One is embryonic stem cells, which is what we work on in the lab, which is what I'm going to talk about briefly. The second are tissue-specific or adult stem cells. Adult stem cells are found in every single tissue type and organ type in your body, and the major function of this is to repair and maintain that tissue or that organ to take care of the wear and tear which happens on a daily basis. And the third cell type, which some of you may have heard about, are induced pluripotent stem cells. These are engineered stem cells made only in the lab, not naturally occurring, and they are created by post-expression of certain transcription factors in differentiated cells. And you culture them in a way so that you produce stem cell cells. So this is just again a repeat of that, to summarize. You have the fertilized egg, you have early stages of embryos, you have a blastocyst, you need to isolate the cells from the inner cell mass and maintain them in culture. These are embryonic cell cells, and these can, in response to specific cues, produce all the cell types in the body. So the major question in the stem cell field is what controls the acquisition of stem cell plate by an embryonic stem cell? In other words, when an embryonic stem cell receives a signal to differentiate, how does it know whether it should stay as a stem cell or it should differentiate? And then furthermore complicating the question, when it differentiates, how does it know whether it should become a skin cell or a neuron or a muscle cell? How does it make these decisions? What happens within the stem cell? In other words, if you were to sort of give stem cells sort of a human life, what does stem cell feel when it receives a signal? How does it interpret the signal? Now, research over the last many years has shown that there are a number of genes which are involved in transcription networks which regulate this transition. There is the control of genetic elements, which is epigenetic modifications, which of course control whether the gene is turned on or off and when it is turned on. And in addition to that, the whole structure of the cell cycle is very different when you look at an embryonic stem cell when you look at a differentiation. These are things that people have known over the last 20, 30 years to be really important in controlling whether a stem cell stays a stem cell or it differentiates. But what we have seen and what a few other labs have seen over the last 10, 15 years is that processes which are involved in classical cell biology, which will appear only in the cell biology textbook and don't appear in the stem cell biology. Processes which are involved in the trafficking of molecules from the outside the cell to inside the cell. And these are typically you refer to them under the banner of endocytosis. We found that endocytosis and the genes involved in endocytosis play a very important role in regulating whether a stem cell stays as a stem cell or whether it decides to differentiate into a specialized cell. So let's try and understand a little more about this. So what exactly is endocytosis? Very, very simply put, endocytosis is the uptake of molecules, typically from outside the cell in membrane-bound vesicles, which are then delivered to the cytoplasm. And once these molecules are within the membrane, the molecules can either be trafficked back to the surface of the cell, or they can be shunted to another compartment so they can be degraded, or they can be sent to different locations. So this whole idea of taking things from the membrane in a regulated manner, this is not just random, it's not, let's just continually take up things. It's a very regulated manner where you sense the signal and you decide how you're going to respond to it. So the work I'm going to talk about today was work done by a graduate student called Nan in collaboration with the faculty of Arts and Pune, Rajan. And then we decided to ask the question, which pathways of endocytosis actually regulate the growth systems? Now, as you may have read, there are many pathways of endocytosis. There's fat-mediated endocytosis, non-fat-mediated endocytosis, granular-dependent, there's phagocytosis, there's phenocytosis, there's lots of other things, dependent, etc. So the question was, if we know that endocytosis is important for a stem cell to stay a stem cell, can we at least understand which of these pathways is really important? Or is it just generalized eating and drinking that the cell needs to do? So the way we decided to do this was to carry out an siRNA-based study. I apologize, I did not put in introductory slide to siRNA, but we can talk about maybe about what you know about siRNA is a small interfering RNA, which through complementarity to the messenger RNA will bind and then will get chopped up by an enzymatic complex, which will then cut the mRNA into smaller pieces, thus destroying the mRNA. So in a sense, if the mRNA is not there, the protein is not going to be produced, and this results in a lockdown of the gene. So what we decided to do was to make a list of endocytic genes which are there in mammals and to systematically lock down each one and ask what is the effect on stem cells when we do this. So if the removal of this gene affects the properties of the stem cell or not. And the way we did this was a simple colorimetric assay. So stem cells have this enzyme called alkaline phosphatase. And the amount of alkaline phosphatase that is there, so for alkaline phosphatase, the enzyme you can provide a substrate and you will get a color. And the color that you are looking for is red. So the redder the cell, the more stem cell line it is, the less redder cell, the less stem cell line. And we are going to check this after putting in the siRNA against a gene which is involved in some sort of trafficking, and we are going to do this in embryonic stem cells. Okay? So we did this for 112 genes, where we had cells plated in these multi-well plates, and each well received an siRNA against one gene. Then we are letting them grow for a bit, and then we are doing this colorimetric assay and asking, have the stem cells changed? We had a number of controls to check to make sure that the stem cells which were not receiving siRNA were still stem cells, that the media was compatible, that the transfection conditions did not affect the cells, etc. And what we got at the end of this is a graph like this. Now, how do you read a graph like this? Right? So, along the x-axis are all the different genes which we have targeted. I've removed the names, otherwise because it's just a series of tiny letters anyway, but I'll talk to you about them. Along the y-axis is basically the z-score, which is your confidence in terms of how different the readout you are getting is compared to the controls. So here, bunched up at the left end of the graph are your controls, so cells which are the control cells and cells which are actually stem cells. And the two red lines basically enclose the controls. Anything which goes above the red line indicates a gene which may be knocked down, resulting in increased stemness. Anything in blue, which is below the two red lines, indicates a gene which may be removed, reduce the stemness largely. So as you can see, we got a few reds, a few blues. Some of these reds have been previously published by other groups, which made us feel very good, but we were actually doing something which made sense, which other people had looked at, and we want to have something completely crazy which no one had seen or thought about before. But the one in blue which we were really excited about was this one, which was the greatest amount of decrease in stemness in stem cells when we remove this gene. And this is a gene called CLTC. It's not a new gene, we've all heard about it. It's the gene which encodes for clathrin. Okay? So, what exactly is clathrin? So, clathrin-mediated endocytosis is sort of the workhorse of endocytic processes. We've all read about it. It's always used as an example to teach endocytosis and the clathrin heavy chain, which is encoded by the gene CLTC, basically forms a part of this structure called the trisphere. So 3 molecules in pink, which are the clathrin heavy chain, and 3 molecules in green, which are the clathrin light chain, and form this 3-legged structure called the trisphere. And this trisphere, I don't have 3 legs. So 3-legged structure, basically it polymerizes to form this basket-shaped structure, exactly like how the wicker baskets are made. For those of you who play football, if you look at the football, it's always hexagons and pentagons. In biology, hexagon assembly gives a fact sheet. You see the beehives, you see there are a lot of other standard structures. The moment you put in a pentagon in the middle of a hexagon, you can curve it. If you just have hexagons, you can't curve it, you get a fact sheet. Every football, there is a ratio of pentagons to hexagons, go count if you have it done so. It's the same ratio which is seen in clathrin codes. If you incorporate a pentagon in the middle of the hexagon, you can increase the curvature, otherwise you will get a flash. So it is through these collateral structures which assemble like this. These assemble around the region of the membrane, which from where you want to take up certain power. So on top of the membrane, you have these receptors, you have ligands binding to them, and at the bottom of the membrane, you have these adapter complexes, which get recruited to receptors, which have a ligand bound. And then at the bottom of them, you have this clathrin assembly happening, which then forms a clathrin protein recipe and takes substrates or takes compost into the cell. So here is another view of the same thing. So slightly simplified. Here are the receptors, here are the clathrin structures. You have this, it also has an actin coming in to provide force. And this then results in the formation of the vesicles, the clathrin vesicles, and the molecules get trapped. So what we saw was that in embryonic stem cells, when you remove the clathrin region, the stem cells start to differentiate, they no longer retain properties of stem cells. And this is how we do it. We looked at alkaline phospholipid staining. So these are wild-type cells on the left, and these are cells which don't have clathrin and don't have another molecule which is involved with clathrin mediated endocytosis. So you can see firstly that the cells no longer form colonies, they are more dispersed, they are not forming these kind of colonies, and they have alkaline phospholipid activity. We also looked at stem cell markers, and you can see that compared to the control, which is shown in the dotted line here, all cells either did not have clathrin, which are shown in black, or another, or a mutant form of another protein involved in endocytosis, all have increased amounts of stem cell markers and have increased amounts of differentiation markers. So this then indicated that the removal of the clathrin region forces a stem cell to leave the stem cell state and start going towards a differentiated state. So we did a lot more work to try and identify which molecules are getting endocytosed, where they're getting trafficked, etc. And for those of you who may be interested, we found that a molecule called epigenetic, which is involved in cells in the region of epithelial line cells, normally gets trafficked by the clathrin-mediated pathway and gets recycled back to the membrane. At the same time, another signaling molecule, which is the TGF-beta receptor, it gets targeted for degradation. Now, when you don't have clathrin, these signals are reversed, and that results in a change in the fate of the cell, pushing it towards differentiation. Now, having said all of this, what you all have also read in all your standard cell biology textbooks is that most cells have endocytosis and it's clathrin-mediated endocytosis. So, complete rubbish, no? I'm saying that you remove clathrin-mediated endocytosis, the cell becomes a differentiated cell. But that's not what I'm saying. What I'm saying is for a cell to transition from a stem cell to a differentiated cell, you need to temporarily reduce the amount of clathrin-mediated endocytosis, which then allows that stem cell to explore other differentiated states. And once the differentiated state is there, it can set up clathrin-mediated endocytosis. So peculiarly, if you think about it, this is how it would look. Like in embryonic stem cells, you have high levels of clathrin-mediated endocytosis or CME. When you reduce it to a certain level, there is an specific threshold. And this now allows the cell to explore different other states. And once it has reached a final state of differentiation, it can re-establish clathrin-mediated endocytosis. So this is what we believe, that endocytosis allows the cell to sort of modulate signals and modulate rates and decide what it wants to do. Do you know how much time I have?
And half an hour more is there.
Good order. I don't think I can talk about it, but yeah. So I'm going to switch gears a little and move a little away from stem cells, but still stay with the same topic of clathrin-mediated endocytosis. But now we are going to switch to a different state, which is a disease. So what I was talking to you about before was early development, which is what we already identified. Now let's say you have formed your organism, and in conditions such as neurodegeneration, where you see degeneration of structures in cells in the brain because of cell death of certain cell types in the brain, what happens, and is there any mode of trafficking or endocytosis for anything? And I'll tell you in detail why we think so. Okay, this is a bit, I think, not with resolution, bear with me. What this shows you is how a normal brain looks. And when you have degeneration, you can see that there is this shrinking of the brain and loss of certain structures which are normally seen in the brain. And this is accompanied by a lot of things, including neuronal cell, there's inflammation, there's effects in the DNA and RNA, there's altered energy homeostasis, there's cytoskeletal abnormalities, synaptic neuronal networks, and a lot of this is largely triggered because of protein activation. So pathological protein activation. And I'm sure you've read about a lot of this also, you have this activation. Alzheimer's, Parkinson's, Huntington's, frontotemporal dementia, just to name a few. So at the center of a lot of these disorders are these aggregates of proteins which are abnormal, which are mutant or which have an expansion of a certain part of the domain, and all of this results in toxicity within the neurons which are created and death of the neurons which are in neurodegeneration. So specifically, I'm going to take the example of Huntington's disorder. This is a genetic disorder, 100% genetic. It is driven by the presence of the mutant Huntington gene, where there is an expansion of the CAG codon. So CAG encodes for glutamine, and this exists within the first exon. And so usually there is an expansion of this CAG codon for a variety of reasons, because of splicing, slippage, all sorts of things. And because of this, when the number of CAGs or when the number of glutamines crosses a certain threshold, usually 34 or 36, you start seeing toxicity, which then results in neurodegeneration. And the phenotypes are very, very severe. There's severe chorea, which is uncontrolled movement. You have a lot of other severe problems in patients who have this. So, what I'm showing you here is in humans of chromosome 4 is the Huntington gene, and when you have these CAG repeats in the first exon exceeding the limit of 35, you start getting these aggregates which form, and the neurons start dying, you start getting degradation of structures such as basal ganglia and an enlargement of the ventricles. Now, there are many models across the world to study Huntington's. Cell-based models, iPSC-based models, mouse models, rat models, all sorts of models. Now, the model we chose to look at are fruit flies. And I sort of want to use this talk to give you an example how you can use sensors in mice, in humans also, and also use fruit flies, depending on the type of gene you're looking at, the type of process you're looking at. A lot of these things are evolutionarily conserved, and depending upon the type of question you ask, you can always adapt the model to suit your question, right? So there's no hard and fast rule that if I train as an embryonic stem cell biologist, I have to stick to it. So you can always find what is going to best suit your question and pick the type of model for that. Now, having said fruit flies, you may ask whether is it even a good model? It makes no sense. We've shown us all these neurodegeneration of the brain of humans, of fruit flies. So let's show you that there is data from the past from other labs which shows that you can use the fruit fly as a good model system, and I'll build on that. The first thing is that fruit flies which contain these pathogenic disorders or these pathogenic repeats of Huntington, do they even show any of the phenotypes which humans do? First phenotype, reduced lifespan, let's look at it. So here, boxed in red are fruit flies which are expressing the mutant form of Huntington. This is a Huntington gene which has 138 CAG. So it's going to encode 138 mutants. So 1 switch is 138. And these are the controls. And these are the controls here on the right. So you can see on the x-axis is basically your days that the fly is surviving. On the y-axis is your viability. And what you will immediately see is that fly which express the mutant form of Huntington, they drop red much earlier. The second thing is that the protein even form aggregates, the same thing as you will see in human patients. So if you have a mutant protein, if the cell is correct, and if the environment of the cell is correct, it should form these aggregates. Answer is yes. So here at the bottom, in pink, you can see these aggregates which are formed by the mutant form of Huntington compared to what is shown in the top panel, which is the control form of Huntington, which has a reduced number of polyglutamine repeats which are non-toxic. So there you can see that the expression or localization of the protein is diffused, it does not spread all over the place, whereas here it is aggregated and it is present in the cell. The third thing, sorry, is do you see neurodegeneration? One way to look at neurodegeneration in flies is to look at the neurodegeneration of the photoreceptors. So this is a great way to look at neurodegeneration because the neurons are dying, which is what you want to see, but it's not affecting the other behaviors of the fly. It's not affecting movement, it's not affecting survival, it's not affecting eating, feeding, moving, etc. Other things which are necessary. So here you can now express the mutant form of continuity specifically in the eye and ask whether it has caused neurodegeneration or not. For those of you who would have seen a compound eye of the plant, usually it's bright red because of the presence of the photoreceptors, and you can see that when we use mutant form of continuity, the eye is completely destroyed. There's absolutely nothing left in the eye. We also established a Drosophila-based cell model where you can see the aggregates inside these cells. So now, using all of these, we've shown you that flies which express mutant forms of huntingtin, they die earlier, they show aggregation of proteins, they show neurodegeneration. So this is a decent system to start asking questions as to what happens to endocytosis in these animals. Why did we want to look at endocytosis? So there were reports in literature which showed that when you look at ligand pathway, in the presence of the mutant form of the protein, there is reduced uptake of other lipophilic bags from the membrane. So here are the controls where you can see in red, there is lipophilic bag entering the cells. But here when you have the mutant form of Huntington, you don't see the entry of the lipophilic bag. Secondly, when you look at the uptake of mutant, when you have these mutant forms of Huntington which are expanded CAG-UP, you get a reduction in the amount of ligand. So first thing, what are the cells we are using? Using Drosophila cells isolated from the larva. These are genetic lines which have fluorescently tagged proteins. The protein we are using here is a clathrin protein which is tagged with GLP so that we'll be able to see the live movement of the vesicles. Okay, so we put it on a dish, culture it very briefly for maybe half an hour or 1 hour and look at it under the microscope. So, first thing, what you're going to see are cells which are, I'm going to interchangeably use wild-type cells and non-pathogenic forms of HPV. Okay, they are both essentially the same. And then you will see pathogenic forms of HPV which have a 138 repeats. The green dots are clathrin-coated vesicles, which is because we have taken the clathrin light chain, which is tagged to GFP. These are live cells, you will see live movies. The first thing you see is in wild-type cells, you see this beautiful movement from outside to in of vesicles moving into the cell, right? Now, when you have a mutant form, and I'm not showing you where the aggregate is, it will come up in some of my slides again, but the aggregate is a big dot, either one dot or many dots inside the cell. So you're going to ask how the presence of these affects the movement of clathrin proteins. Now the video is actually playing. So you can see the difference between left and right. When you have a mutant form of Huntington, these vesicles are being moved. They don't leave the membrane. Wherever they are in the cell, they just sort of sit there and blink. There is no two things to notice. One is here, all the movement is directional. It comes from outside to in. It always goes along specific tracks and it's going at a certain speed. In this one on the right, it's just staying there. And maybe just doing this. That's it. But staying in the same place. Really no movement, really no directionality, just doing this, no useful movement. How do we quantitate this? We quantitate this with what are called kinographs. So kinographs are basically you just take 1 point or 1 pixel or 1 vesicle and you plot its progress over time and distance. So along the y-axis is time and along the x-axis is distance. So if we start at a certain point with this vesicle, you can see that over time it moves on. You follow my pointer? Similar. Here you can start here and you can see it moves on. Now, in the case of the mutant form of Huntington, over time, when I move, it's just staying there, it's not going anywhere, right? So this is the way of quantitatively showing that the vesicle is really not going anywhere. Then we calculate the speed. Speed here is a negative unit, which you might find a little strange because there cannot be a negative speed. What it basically indicates is you are measuring the outside from the periphery of the cell to the inside. Inside is considered as zero, so anything coming from outside to inside is just negative. Okay, so you can see that from outside to in there is a certain speed, but in the case of the mutant Huntington, it's just a zero. It just doesn't move, it just stays there. Now, this is very interesting. This is to mark the direction. So these are vector coordinates. So circle is 360 degrees. Now, when you looked at the videos, they were always moving along the radius. So 180 degrees is what it was moving at in wild-type cells. So it's all 180 degrees. Every single one of those spots is always moving towards the inside of the cell in a straight line. These guys in the mutant pathogenic forms, like I said, they are doing this, they are moving anyhow. So they are covering the entire 360 degrees and going nowhere. So it's basically useless movement, right? Just standing in one place and moving like this, but going nowhere. So then we asked, okay, is the uptake of lipids also truly affected here? So you're seeing that there is a defect in the movement of catholic vesicles. Ideally should have lipid inside them. So if we measure the amount of lipid taken up in our system also is there a reduction. So we use a lipid which is marked here in red. You can faintly see it in the wild-type cells, but let's look at the quantification on the right. So in wild-type cells, you have a large amount of lipid taken in, and that amount is hugely reduced when you have the pathogenic forms of Huntington. So not only is movement of catholic vesicles affected, but the uptake of lipid is also affected. So, one of the things on which clathrin-coated endocytosis depends is actin. So, imagine this is your cell, this is your plasma membrane. Now you start holding the plasma membrane. At some point, you're going to need some force to pull it in, right? That force initially comes from actin. And I think you've all read about actin, right? So, actin is this polymeric form of a cytoskeletal protein, and there are workers which walk on it. So what happens is at the area where the cell membrane is starting to thin, actin starts ascending, and then it starts creating a pull. So it's almost like you would have seen at construction sites, they pull, and if that pull is not tight enough, nothing is going to move. So we asked, since there is really no movement in these pathogenic Huntington-containing cells, is the actin also disrupted? We've seen that the lateral movement is affected. Is the actin also affected? So here, again, we're using Drosophila cells, larval cells. Here the GFP is because we have marked protein which binds to actin. With GFP, we are implementing actin. So here on the left are your wild-type cells. Here on the cells are cells which express the pathogenic forms of Huntington. And when you look at wild-type cells, you see really gorgeous streaming movement. There's so much dynamicity of the cell. The cells are really not fireplace. There's a lot of stuff happening inside them. And when you look at your cell which contains pathogenic forms of Huntington, you can see that it's so different, right? So we still have to figure out what to say. Is it less? Is it more? We just say it's different because the actin seems to be organized completely differently in these cells. So we've seen that fraction is different, uptake of lipids is different, actin is different. So we can ask, can we quantitate the actin flow? And all I want you to look at is this right side thing, the color coding. The redder you are, the more is the actin flow. The cooler you are, the bluer and the blacker you are, the less is the actin flow. And it's really dramatic, the difference between wild-type cells and cells which contain these Huntington aggregates. In the wild-type cells, you can see that close to the cortex of the cell, there's so much actin flow and movement, which you saw about as spiky philopodia come down. And in the context of Huntington, pathogenic Huntington cells, it's really not going to be typical, right? So let me just explain this to you and then I'm almost at the end of it. So we see the original of clathrin-mediated endocytosis. You have receptors, you have clathrin. And here now are these connecting proteins, which help to connect clathrin to actin. Because otherwise you need a bridge. If you don't have a bridge, you can't connect the two of them. And that bridge is a protein called KIT1, which is shown here in purple. So we then asked, is there some problem with this connection between clathrin and actin when you have these pathogenic forms of cancer? And in this fitness, so what we did, this was in LSD. So let's come here. So we asked and we found that when you remove that connection between actin and clathrin, lots of things get disrupted. Clathrin gets disrupted, actin gets disrupted. But we asked in the context of pathogenic Huntington containing cells, if we were to try and put more of that connection between clathrin and actin, can we now rescue clathrin movement? Can we now rescue actin dynamics? And can the cell be made to believe that it's in a happier state? And so we did that. We overexpressed HIT1, which is this connecting protein. So again, here on the left, I'm going to show you a video. So these are again cells which are expressing clathrin tagged to GFP. So you're going to look at clathrin protein vesicles. And on the right are the same cells. And what I want you to see here in the inset in red are the Huntington aggregates. So we have not removed the aggregate at all. The aggregate is still there in the cell. We have just provided more of the protein which connects clathrin to actin. And suddenly you can start seeing that there is this movement of vesicles from outside to in. You no longer have this type of distribution. We can see that the tracks are also re-established. We did a number of other assays also to check that it was indeed almost restored to normal. Now, what is, okay, so I'm going to take a little side step and then come back to the main story. So remember I told you that how would you feel if inside you there was a traffic jam? You were a cell and inside you there was a traffic jam. And you would sort of feel like everything is blocked, nothing is moving, everything is sluggish. So the question then becomes, is this aggregate inside the cell, is it working as a physical entity or is it working as a chemical entity? In other words, is it just a big stone in the middle of the road or is it actually the police wala who say, aao license karo? What is it doing? Is it just creating molecular interactions and specifically sequestering proteins that they cannot do their normal function or is it just sitting there and just blocking all kinds of words? And how does a cell feel when it happens? So we decided to work with a physicist at ICER and we collaborated with Professor Shiva Patil at ICER. He's a physicist and he uses a technique to measure the stiffness of cells. And the way you do this is through a technique called atomic force microscopy. What do you do with this? You take a cover slip to create your cell. On top of the cell, you come with a cantilever. Cantilever is nothing but you can think about it as a sort of a flat thing, at the end of which you have a beam. Now, if this is a cell and this is my cantilever, the cantilever will touch the cell. The cell is fixed, but if the cell is very stiff, then what will happen is that the cantilever will start bending, and that degree of deflection of the cantilever is measured with the help of a laser beam. So the laser beam is positioned on the cantilever and it measures. So it's a direct proportion of how stiff the cell is. If the cell is very soft, what will happen is that it will just dip like this. Whereas if it is very stiff, cell will not move, but your cantilever will move in a different way. So using the deflection of the cantilever, you can measure what happens to the cell. And what we saw was that when you have wild type cells, along the y-axis is measuring in pascals, which is your Young's modulus, which tells you how stiff the cell is. And along the x-axis are the levels of the temperature. So the cell is reasonably soft when it's a normal cell. The moment you have an HTT or a Huntington aggregate in it, the cell becomes stiffer. So clearly, because you have this big thing which will be filling up the cell and making it really hard. Now, we then asked, because we had seen this interaction with actin, we had used this protein to restore actin and clathrin interaction, can we now rescue some of these phenotypes by playing with the way the active cytoskeleton was formed. And to do this, we did two experiments. One, we added a pharmacological inhibitor for only 5 minutes. Now, in 5 minutes, no aggregate will be resolved. Okay? But this pharmacological inhibitor breaks down the active cytoskeleton. And we found that the moment we did this, the cell which contained the Huntington aggregate also became very soft. Meaning, therefore, that the aggregate itself, as a physical entity, was not increasing the stiffness of the cell, but the aggregate, because it was associating and sequestering other proteins, maybe not allowing them to take part in their natural processes, it was changing the internal environment of the cell and making it exceedingly stiff. And just by using 5 minutes of the pharmacological inhibitor, it could rescue the state of the cell. Now, we also overexpress this protein which could rescue the actin-mediated process within the cell. And we found that that also rescued the stiffness of the cell, and it was more or less similar to wild-type cells. Now, of course, the true test of the whole thing is whether it will rescue neurodegeneration or not. All this is very fine, right? Saying all of this, what happens to the neurons. So again, we looked at the fly because the photoreceptors are a very good model to look at neurons without affecting the brain, without affecting the viability of the fly. So we asked what happens when we overexpress some of these proteins. Can we rescue neurodegeneration even though the aggregate is still present? So all of these experiments are done in the background of pathogenic Huntington. Here we are using Huntington, which has 127 repeats. So you see the eye here, absolutely no red pigment, it's completely destroyed. This is control GFP, the eye is still destroyed. When we start putting back elements of the active cytoskeleton, you can see the eye is destroyed, and here is our favorite protein, which is used to develop calcium active, and you can see that the eye is destroyed. So what we see is that even the presence of the aggregate, you can now find a way to restore neurotoxicity, which then also restores endocytosis and allows the active cytoskeleton to arrange itself normally. And these may now be new ways to think about coming up with therapeutics for neurodegenerative conditions. So to summarize, what I've shown you is that in the presence of these Huntington aggregates, which are here, you have stored endocytosis. Endocytosis really doesn't move, you don't get release of vesicles from the membrane. But the moment you either modulate the active cytoskeleton, either through adding certain proteins or using pharmacological inhibitors or overexpressing HIF1, you can now rescue endocytosis and you start getting the release of these vesicles from the membrane, and you can also reduce neurotoxicity under such conditions. So I will stop here, but not before thanking the people who actually did all the work that I spoke about today, and all the funding agencies. So this is Surya who did the second part of the work, and these are people who did the lab from some time ago. These are all the people I collaborated with, Rahul and Shiva, various projects, Amrita and Chetan, and these are our sources of funding. And I would thank you all for being very outstanding on this. Thank you.
Thank you, ma'am. Any questions?
It was a wonderful talk, ma'am. Thank you so much. My question is that one is the therapeutic part. So this was done at the lab. Has it gone to the clinical trial or
No, not yet. So we're still trying to understand more about that interaction with body. But you're right, these are things which can potentially play a role in further therapeutic intervention. Thank you, Minister.
Thank you. Good morning, ma'am. Thank you for the wonderful talk. My question is that the clathrin-mediated pathway that you talked about, can we use that in antiviral therapies?
Absolutely. That's what. Okay. So clathrin-mediated endocytosis play a very critical role in treating uptake of viruses. The problem is it's tough to target the entire pathway because it is present in every single cell. And if you block the entire pathway, after a while the cells will die. So you may need to find specific adapters which are involved in uptake of only the virus, and that can be something one can try to target or attack or block for specific viruses. But if you block the entire pathway, you will have a dead organism.
Anyone else? Very nice talking to you. I was wondering if in the last part of your presentation you showed how by making those modifications for the movement. Does this have some positive effects on the cell with respect to the aggregate also? Will that reduce the aggregate accumulation in the cell or something?
That's a great question. We haven't done it over a long enough time period to see whether the aggregates are taken away or not. At least in the case of HIF1 overexpression, we find that the aggregates are still there. So they don't affect the aggregate, but they allow the cell to carry out its normal functions in a reasonable way. So, one will have to sort of add certain other things which will also over time reduce the aggregate while also restoring the normal functions of the cell. Thank you for the question.
I just wanted to know if you told me that it can be open to one government server. You told us about it can go on one disorder, but can you tell me if it can go on Parkinson's or paralysis?
So, very interesting question. So, I didn't show you these parts, but we looked at 5 other disorders which are triggered, 5 other neurodegeneration disorders, including Alzheimer's. We did not look at Parkinson's. No, we looked at Parkinson's also. And in those, that's immediate endocytosis is not effective. However, when we look at a certain type of frontotemporal dementia, where another protein is mutated, TGP43, there it's actually mediated endocytosis of the victim, and there maybe similar mechanisms going on. So what this tells you is not all neurodegenerative disorders are the same in terms of affecting endocytosis and actin oxidation. Only some of them affect endocytosis and actin oxidation. And Huntington's happens to be one of them. So I think in the case of other disorders, such as Parkinson's or Alzheimer's, one will have to look at other molecules and other pathways which may be affected, and those can well be a way to intervene therapeutically to help.
Thank you so much.
Thank you so much, ma'am. It was really a wonderful talk. And we are very grateful that you have spared your valuable time and shared your information with us. And now we have 10 minutes break. And then the last session.
Thank you.
1 मिनट से ये ज्वाइन करते हैं
या वो आप ज्वाइन कर लीजिए.